This is episode 82 of the Immunology Podcast, Immunological Memory with Dr. Susan Kaech. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Susan Kaech from the Salk Institute on the podcast to talk about her research on memory T-cell development. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
STEMCELL Technologies would like to introduce you to the Human Immunology News. It covers everything from immunotherapy, autoimmunity, and adaptive and innate immunity. Human Immunology News keeps readers current with the latest news, research, policy events, and jobs relevant to the immunology community. Subscribe for free at www.humanimmunologynews.com. Hi, Jason.
hey.
It's been a while.
I know, I'm too excited here. We got nominated for something.
We did? For what?
Yeah. Apparently a People's Choice Podcast Award. And so you can go vote for this
No way.
by the time this comes out at podcastawards.com. Vote for us in the science category.
Very nice.
It's exciting.
I hope we win. We need our listeners to step up. So go for it. So I know that Jason is very excited and he really wants to have a prize. So he only does it for the recognition.
That's true. It's hard for me to post on socials otherwise.
Very nice. I mean, and then what else? Next week. So this week we are also celebrating another win.
Oh, America's win? When we
250 years?
Independence Day will be the week this comes out. Yeah, so, the usual fireworks for the family. I don't know if we're doing anything else. I also have all next week off as a summer shutdown for my company, so that'll be kind of nice. I can recover from my not vacation.
Yeah, I heard it was a little bit warm in your LARP camp.
It was, and I indeed had to provide a very large amount of out-of-game medical care in an unanticipated and unprecedented manner.
Oh, my. Well, I'm glad that they had you there to take care.
Yeah, it was good that I was there. It was a little hairy, but we got through it. But I learned a lot of wilderness medicine and how to drop an IV in a cabin and then hang it with a coat hanger up from the ceiling rafters, things like that.
Oh, okay. That sounds serious.
Yeah, it was real medicine. It wasn't just boo-boos.
Oh my. Well. All right. So, it's been a while since we'll see each other. Why don't you tell me what papers you brought in for today?
Boy. So I'm going to start with a paper that came out in Nature, which you're like, why is this in Nature? But it's actually a very interesting paper on bacterial function, which is exciting and weird and different. So it's in Nature. It is called an alternative broad specificity pathway for glycan breakdown in bacteria. First author is Saeed Amarahosian Nasseri, and last author is Stephen Withers. Came out June 19th. Was received in June of 23. So, the usual, that's not bad for a 1-year review time on Nature and the turnaround. So high level here, there's something called Koshland mechanisms, which is how most glycosidases work. What are glycosidases? They chew up cellulose and things like that, right? Cellulose is super stable. Normally takes millions of years to break down. Enzymes make that much more efficient. We know bacteria can chew down cellulose. Highly evolutionary conserved, high specificity in these enzymes for very specific things. But there isn't enough genes in the bacterial set of genomes to handle all of the different these glycosidic bonds that exist in nature if you just have only these specific enzymes, right? So they've always known something's up, but haven't been able to figure it out. So they find something called non-Koshland glycosidase activities for the first time and then map out the mechanism therein. And these enzymes break out both alpha and beta stereochemistry and multiple connectivities. And they also cleave substrates that are not hydrolyzed by the standard ones. So they're actually more promiscuous. So, and they see homologs of them in both gram-positive and gram-negative bacteria. So they basically take some phosmid libraries. So these are the F0, F1 giant plasmid types of bacterial libraries. And, pump enzymes out of those so they have transcription occur, and then try to figure out what can break down these fluorescent versions of glycoside substrates to screen. And then they find some bacteria that do it in the gram-negative land. And then they spend a bunch of time mapping out what these 5 enzymes do, AL1 through AL5. They figure out where they are. They get crystal structures. They map out the substrates. They do NMR to figure out the stepwise function. They figure out that they have to have some type of redox substrate, so they work very slow unless you're regenerating the NAD, the NADH. They isolate that. They don't figure exactly how that works in vivo. They have to do it ex vivo and use an artificial substrate. But they basically map out the whole kit and caboodle. They map out where they are in the genomes of some of the bacteria. They see homologs. They see homologs in the gram-positive bacteria as well. As I said, X-ray crystal structures, and then do a substrate screen and see that they have broad substrate specificity, so much more broad than typical. So the point of all this is they are able to identify for the first time and isolate down to enzymes the way bacteria can break down these very common plant matter substrates that are in the human gut is introduced to all the time, to understand what's going on there, if that makes sense. It's quite fabulous in terms of understanding this. They also point out that some human drugs have this linkages in them, and these could be a way that the body is metabolizing drugs, or the microbiome's metabolizing drugs. So why is it a big deal? We knew it happened because duh, but we never figured out how. And so we've been able to finally isolate how bacteria do this major activity that we already know for some of it. And they don't just kind of map it, they map it down to the organic chemistry bond. So I'm not going to go into all the organic chemistry bonds here because that is very not fun to describe verbally. But it's a redox reaction, so it'll hydrolyze it by attacking the oxygen typically and breaking it there and then reducing it afterwards again. So oxidate, reduce. And there you go.
I very interesting. Although I didn't know we now have the microbiology podcast.
So yes, this is a little bit different. But it is fundamental to how all our bacteria work, and it looks like it degrades a bunch of drugs for everything and its uncle. And it's a little microbiology this time. Not every week is straight B cells or T cells, but remember, macrophages love bacteria.
Yeah, I guess it is up your alley. Anything,
It's also like so fundamental to
Yeah.
the microbiome, and we don't know how this works, but like we obviously degrade all these products in our gut. And
Yeah.
that is really important, and those metabolites affect our immune system, but no one was able to understand how any of this worked. It was a giant black box.
I'm actually kind of impressed that we didn't know. Feels like something so fundamental that it would have already been figured out.
No, it's cool. They finally got there.
All right. Why don't I continue? This story was actually a contribution from a listener. So one of the co-authors in this work. I really like it when people kind of send over their work, and sometimes it's really nice and it really fits very well with Roundup. So feel free to send to shamelessly plug your papers to us. We might just take up on the offer. So this paper is called Risk of Second Tumors and T-cell Lymphoma After CAR T-cell Therapy. And the first authors are Mark Hamilton, Takeshi Sugio, and Troy Norderbos. And this is from the lab. So the corresponding author is David Miklos from Stanford University. And this paper kind of took me down a little rabbit hole of the current status of CAR T-cell therapy and safety and regulation, because CAR T-cell therapies has been in the clinic for a while. It has been officially approved by the FDA and by the EMA some time ago already, a few years ago. So it's actually, becoming more and more common to use it as second line or like one of the lines of therapy against multiple leukemias. And something that has become evident now that there's enough patients treated with CAR T cells when it comes to the whole monitoring of the safety of this therapy is that many patients, after successfully responding to CAR T cell therapy, actually develop new cancers. Oftentimes, they will develop some kind of other, blood cancer or even some solid cancer, some solid tumor. And this, uh, when there's a few, especially last year, there were a few studies, kind of meta studies that show that, for example, of a set of over 12,000 CAR T cell therapy treatments, over 500 resulted in a secondary malignancy reported to the FDA as part of the monitoring of this therapy. And this is about 4.3%. And people are, of course, as with any therapy, especially something as kind of, um, what would be the word? Genetically modified therapy. People are get very anxious about potential oncogenicity of the genetic modification, and so where are these cancers, are these tumors coming from? Um, are the genetic engineering of these cells does this have anything to do with the appearance of the secondary malignancies? And so this paper is part of a series of studies. So there's other similar publications that kind of try to shed some light on, from these secondary malignancies that we observe, which ones, if any of those, can be kind of adjudicated to the therapy. So let's just point out that there has been a couple of examples already well known of uh, car integrations, like the genetic integration of the car, having a particular impact and having kind of a effect on the T cell product itself. And I think the most famous one, which was shown I think it's 2018, was this uh, patient that had a particular clone of T cells kind of overriding the whole CAR T cells after infusion, because this clone had a disturbance, had a disturbance on the TET2 gene, and this actually uh, was a way of kind of was how these researchers, they actually realized that TET2, basically knockout of TET2, increased the performance of these T cells, and therefore it was not a malignant transformation. The T cells did not, the patient did not suffer from a tumor, or did not suffer from, was not a negative thing for the patient. But you can see that the risk of negative malignant transformation was not, couldn't be ignored. So in this case, this particular paper, so in this work, what they did, they look into 791 therapeutic CAR T cell infusions of patients treated at the University Medical Center at Stanford University. And then from these patients, 25 had actually presented with a second primary tumor. But of these ones, only 14 were hematologic. The rest were solid tumors. So people also often develop melanomas, carcinomas, different type of carcinomas. There was also a patient with a mesothelioma. And from the 14 hematological cancers, only one was actually a T-cell lymphoma. And this is a kind of events that really catch the attention of the authorities, because where is this T-cell lymphoma coming? Are these T-cells that make this lymphoma related at all to the T-cells that were infused as part of the CAR T-cell treatment? And this is an example of how people are analyzing this kinds of secondary tumors to evaluate whether the CAR T cell therapy can be, what is the role of the CAR T cell therapy itself. So this person had a diffuse large B cell lymphoma. She was treated with these axicabtagene uh, ciloleucel. I don't even know how to pronounce this. So Axicell, aka Jascarta. This is the CD28, the CAR T cell against CD19 that has a CD28 co-stimulation domain. And what they observe is that after kind of a successful uh, initial clearance of this CD19 positive cells, they observed that the person developed a T cell lymphoma. This patient, one of the things to keep in mind, this patient was EBV positive, so the B cell lymphoma was probably a consequence of the Epstein virus infection, and this T cell lymphoma was actually also positive for Epstein virus. And so what do we do? So what did how do you look into this new T cell lymphoma? They find that in fact it was EBV positive, although the patient, they hadn't detected EBV in T cells before the infusion, but then so apparently somehow they got infected later. They do see that there's a particular TCR beta clonotype of these T cell lymphoma, which was present in the patient before the infusion. And this lymphoma was accompanied by an increased viral load of EBV in the patient. So really, it seemed like EBV had something to do with the transformation of these T cells. In the end, what they could not detect was mRNA of the CAR, of the chimeric antigen receptor itself, or any CAR DNA within the genome of these cells from this T cell lymphoma. And here's where the kind of really, I think the most interesting part was that what they see is that some of the founder mutations that were found in the original B cell lymphoma, and this is particularly TET2 and DNMT3A, which they were probably part of the initial mutations that allowed this B cell lymphoma to develop, were also found on the T cells. And they suggest that then these two mutations were originally in the common progenitor, in a lymphoid progenitor, up in the differentiation cascade. So what they agree on is that in this case, although it is a T cell lymphoma, it is not a consequence of the genetic modifications of the CAR T cells. It's just because it's probably caused by the treatment itself and the perhaps the on the one hand the lack of the chemotherapy, the preparing chemotherapy for receiving the CAR T cells, maybe that you know uh, really weaken the immune system. The absence now of B cells that can make antibody probably allowed this new tumor, this new malignant cell to proliferate, and without proper immune response. And this, I would say, is probably, when you look at all the other reports in the literature nowadays, a lot of people discussing this, I think it is quite, there's very few, I think it's maybe just two examples in which the introduction of the CAR seems to have had something specifically guiding the development of the lymphoma. But in most of the other cases, it's just that these patients already have a heightened predisposition to have a secondary tumor. And oftentimes the application of the CAR T cells with the accompanied depletion of B cells and then a reduction of your immune response is probably the reason why these patients then end up developing secondary malignancies. But it doesn't seem so far that there's very little evidence suggesting that it is the treatment itself and the genetic modification of the T cells that is driving the secondary tumors. That being said, the FDA last year has forced or has made the companies add to the warnings of CAR T cell therapy that there is a heightened risk of developing secondary tumors, which I find a little bit, I don't know, I find a little bit funny, because I think it's probably a little bit misguided. And it gives people the impression that the CAR T cells are a risk for cancer.
Yeah, it was a very aggressive move by them to add it, and people were not particularly thrilled, I will say. But we'll see, I guess. Right. But this is interesting that it seems to be related to just the general treatment or their already existing mutation burden, but not from the drug itself. I'm glad
Yeah.
they're watching it, and it's important work.
Yeah, for sure. I mean, we need to be certain, but I do think that the main reasons is that this, if you develop cancer once, you already have certain mutations that probably, give you an increased chance of developing cancer again. And the fact that you go through all this chemotherapy usually before that those are mutagenic, potentially mutagenic therapies, that is probably also increasing new mutations, that you will acquire new mutations that could, down the road, if you're cured of this first cancer, but then there's space for a second cancer to develop.
It's complicated. Like I just it's it's so new. They threw a warning on it, and then now everyone's having to do a bunch of post hoc work to figure out what that means.
Yeah. Yeah.
So something to think about. Something to definitely think. I don't know. I'm a mixed thoughts, right? About what we do about it. Like, do you put the warning on and then do the investigation? Is the warning too soon?
Yeah. I mean, but also the warning, if you CAR T cell is used in patients that already have such bad prognosis, then the small chance of getting, so this is 3% chance of getting a secondary tumor, means that you survived your current tumor. And I think this risk is irrespective of the treatment. So if you had cancer once, you have a higher chance of having cancer again in general.
Yep. I wonder if that's that
So,
mutation burden notion or not.
hmm.
All right. Well, that being said, let's hop over. I don't have a good segue here other than Hep B causes cancer and there's immune system ties. But anyways, we're going to talk a little Hep B here. I think this is a super interesting story from an immunology perspective, even though it's also an infectious disease. It's okay. All right. So it's in Cell. It's Therapeutic Potential of Co-Signaling Receptor Modulation, Hep B, published June 18th. First authors are Francisco Andriata, Chiara Laura, Nicole Rava, Caroline Kruger, Zena Fitch, Keigo Kawashima, and Kristen Beccaria. Last author is Matteo Iannacone, if I pronounced that right. Apologies, that's a lot of names. I'm sure I have an error rate somewhat worse than DNA replication there when I'm saying this. All right, so that being said, right? Hep B is notorious for not always being fully cleared and sticking around and kind of being a pain in the butt of an infection. And once it's cleared, you don't really know it's gone. We know that there's some dysfunctional killing element of it. And what they do is they use these really fancy mice that have kind of a chronic Hep B model, but then they can prime it on the other side with either hepatocytes or Kupffer cells expressing the rest of the Hep B antigen. So they take Hep B transgenic mice that express Hep B core protein in hepatocytes into native T cell receptor, and you transfer in adoptive transfer of naive T cell receptor with this restricted epitope for Hep B, and then they can prime it, and they can either do instead of a Hep B and hepatocytes, they can do it in Kupffer cells. So what they show is that when you take the T cells with a restricted to region of Hep B and put it in mice that have Hep B in the hepatocytes, which is where it goes, things don't work well, right? Killing doesn't work well. It's kind of a broken program. If they turn off checkpoint inhibitors, it doesn't do anything to fix it. But if you do a 4-1BB agonist or an OX40 activation, you get T cell function to resume, and 4-1BB is better as a potentiator of T cell activity. Okay, so that's a little interesting, right? Like, that's broken. We already knew it was broken. This isn't a surprise, but showing that checkpoint inhibitors doesn't fix it, that's new. Showing that these two agonists work, especially 4-1BB works, that's new. That's cool. That's kind of interesting, right? Well, then they say, what happens if I put it in Kupffer cells? If I put the Hep B core in Kupffer cells, which is not where it goes to normally, right? That's not normal. That's normal for other infections, but not Hep B. All of a sudden, these T cells need no help. The adoptive transferred T cells need no help to go kill the Hep B. And that's what's really cool about this paper, is they show that fundamentally something about the chronic antigen nature of it hanging out in Hep B leads, the Hep B hanging out in the hepatocyte, leads to dysfunctional T cells. They look like exhausted cells to a certain amount on RNA-seq and on other metrics, right? But they aren't. 4-1BB helps rescue them. But fundamentally, it has to do with the fact that the antigen is being presented by the hepatocytes that leads to the downstream dysfunction. And when you put it in a Kupffer cell, which is, the hepatocyte macrophage type thing, it's fine. It's something about it being presented in a hepatocyte that shuts everything down. And that's why you have this dysfunction as chronic infection state. And then they can see this pattern in people as well, in terms of they take the cells from people that are infected, give it 4-1BB, and it kills better in vitro than if they don't. And checkpoint blockades do bo diddly. So I summarized a lot here. It is a Nature paper with figures 1 through 1 million and all the supplementals and videos and not Nature paper, sorry, a Cell paper with all of those things. It's very exhaustive. It's worth a read if you're in the Hep B field or trying to understand T cell exhaustion or how T cell dysfunction can lead to a state. But it's really showing in this case the cell presenting the antigen can alter that response quite drastically. There you go.
Yeah, I guess it makes sense. I mean, hepatocytes, I think that's not completely unknown that hepatocytes are kind of tolerogenic in a way. And also 4-1BB, aka CD137, well known co-stimulatory domain, used also for like CAR T cells, for example. So
Yeah.
I'm not completely surprised, but
Yeah, no, I
I
just thought
think
it was really neat that they are able to overcome it, right?
so. They do, I guess, they do the mouse experience, I assume,
Yeah, they do
and
this on mice for the transgenic.
and they look into the Kupffer cells are tissue resident macrophages in the liver. So are they just all this experience, they do it just with the antibodies, with agonist antibodies, or are they also using some kind of expression, some kind of like genetic model?
This is a genetic model, so like these are genetically transduced to be infected this way. So they force the antigen into the Kupffer cells as a transgenic system.
Okay. No, I understand. All right, I missed that part. Okay.
Yeah, it's
Very nice.
really quite sophisticated. Like, the cool part here is the molecular biology manipulation they did to be able to do the study, and it's pretty
All
neat.
right. Okay. All right. So for my second paper, I'll have to be brief because I took too much time talking about the CAR T cells. I just thought it was very interesting.
Really?
Yeah, I know. I saw you looking at me thinking this girl is talking way too much. I just thought it was very interesting. So the second paper, I liked it because it is a follow up on a paper I presented earlier. Do you remember those experiments, that 10-year long experiment in which they kept transferring T cells from a mouse to another and then stimulating it with antigen and then transferring the T cells to a new mouse? And they kept doing that for 10 years.
Mm-hmm.
So, and what they saw basically, and they already published, is that the T cells kept proliferating. If you gave them enough time to recover between the inoculations, then you can just take the cells from the one mouse, sort them and re-inject them in a fresh young mouse, and the cells would keep going. They also saw that the cells kept better and could be kind of transferred for longer if they were given 60 days between stimulations instead of 30 days, so that giving them a longer time in the end improved the times you could transfer these cells from mice to mouse. And I thought that was very cool, and I really like that story. So this is, of course, coming from the originally from the lab of David Masopust at University of Minnesota, but now we also have corresponding authors Caitlin Zebley and Ben Youngblood at St. Jude Children's Research Hospital. First author Tian Mai, and the paper is called "Conserve Epigenetic Hallmarks of T cell aging during immunity and malignancy." And this was actually published in Nature Aging on the 12th of June. And I have to say, I'm not an expert on aging, but lately I've seen a lot of this whole study, epigenetic clocks and how you have an epigenetic age, and all these influencers saying they have an epigenetic age that is 20 years younger, or silly things like that. But well, so in this case, what they look into is trying to understand how does the epigenetic landscape of the T cells reflect their story. And I think this is very interesting in how it relates to the age of the mouse. Because if you do a regular experiment in which you're measuring, epigenetic of a T cell in one host, you are limited by the lifespan of the host. And because of this experiment, that's exactly a thing they got away, um, kind of pushing aside, right? They could make these T cells live throughout many lifetimes of the host. They could see how the epigenetic life of the T cells looked. So basically, what they did is they analyzed all these methylation patterns, and what they see is that they could find a epigenetic signature that was very unique to the T cells, that changed as with the amount of time that T cells had been kept in this experiment. So you have like different time points. For example, a naive T cell, then a T cell that was kept for like half a lifetime of a mouse, like a year or something. Then when the T cells were approaching the lifetime of the mouse, that's kind of another time. But then you have like multiple lifetimes, which included about 5 mouse lifetimes. And what they see is that they can identify a very specific, like T cells seem to develop a very specific epigenetic aging that is independent of the age of the host, but it's basically just a reflection of the proliferation story of the cells. And this epigenetic changes still continue changing after the 2-year mark of the life of these T cells. And naive cells would consistently keep a very kind of quote unquote young epigenetic age, regardless of the age of the mouse. So really the epigenetic modifications that were associated with kind of aging of the cells really only depended on the proliferation history of T cells. Because we know that T cells also, and as this experiment showed, is that they can keep proliferating for pretty long. They're capable of maintaining their telomeres, and this epigenetic aging was not a reflection of a diminished capacity to proliferate, which are all things that in a way for regular somatic cells, people seem to associate epigenetic aging with a progressively reduced capacity to proliferate further. And there was a lot of course, there's a lot of descriptions of the different genes that are particularly changing or methylated, or for specific regions are methylated as the cells uh, proceed in this experiment. But what is important is that they draw some parallels with malignant T cells and with lymphomas, and how the clocks kind of compare to normal non-transformed T cells. And they really show that these epigenetic changes, although T cells can live many lifetimes of the host, they don't accumulate this type of methylation patterns or epigenetic changes that are seen in proper malignant cells. And this also seemed to apply both for the mouse that we're studying, but also when they looked into human data and compared, human cells and human cancers, that there were specific methylation patterns that were non-malignant that kind of kept the T cells going and characterized their growth, their proliferation story. I think there's a little bit, some of the markers, and other things that there was very interesting to see some of the differences. But I think that the takeaway message from this paper is that T cells really have their own pace, and they have their own mechanisms to keep their epigenetic clocks in their own pace, and they are independent from the age of the host, and they can either stay very young epigenetically, or they can, in the case of naive cells, even at older hosts, or they can age very quickly in the case of cancer of lymphomas, in which you have lymphomas that epigenetically are 200 years or something like that, when but they're present in child, for example. So some of the samples that they looked in human from their human samples.
So you're essentially saying that the mouse is what ages the cells. The cells don't age themselves.
Right. So the cells can keep, like, the main source of epigenetic aging, so to say, or the epigenetic changes that accumulated throughout time, was just the amount of proliferation that the cells had experienced. Because also they saw a difference if they did the stimulation every 30 days or 60 days. 30 days accelerated the clock.
So if you put it from mouse to mouse to mouse, the cells will age even if the mouse is young. So aging is intrinsic to the cell.
Yes.
With its replicative cycles. The more you reproduce, the older you get.
But the T cells, they just kept going. So even though they were epigenetically aged, that did not necessarily correlate with a diminished capacity to survive or to keep proliferating. At least in the case of T cells. Yeah. Forever young. They can live forever, those T cells. They could outlive you if you weren't getting old.
Here you go. Well, we're going to be talking more about T cells and memory here in this moment with Dr. Susan Kaech at the Salk Institute. But before we get to that, are you looking to improve your T cell therapy development, Brenda? With STEMCELL Technologies' new GMP manufactured Immunocolt XF, you can achieve robust T cell expression and expansion with high cell yield and viability without the use of serum. To learn more, visit www.stemcell.com/immunocoltxf.
Joining us for our interview today is Dr. Susan Kaech. She is professor and director of the NOMIS Center for Immunobiology and Microbial Pathogenesis at the Salk Institute in California. And personally, I'm very happy to have Professor Kaech with us today. Her work on immune memory has been, very important in the development of the field. And her lab just, there's so many cool stories are in her lab right now. So I'm really happy to have you today, Professor Kaech. So thank you so much for joining us for the Immunology Podcast.
Well, thank you. I'm very excited to be here. As I was saying, this is my first podcast, so I'm a podcast virgin.
Unbelievable. We're happy to be your first. So we'll be nice. We'll be nice.
Thank you for being here. And Brenda, why don't you ask the first question there?
All right. I just want to, if I may, start a little bit. So first of all, I really like your presentation now at the AAI. I was following with very much interest, so maybe we can go into a story. But I was hoping you can give us a little bit narration of your background. I think people in the T-cell field are very familiar with your work, particularly the work you started when you were a postdoc at the lab of Rafi Ahmed, very big name in immune memory and reference in the field. So I was hoping that maybe our listeners, those who don't know you, can get a little bit of an idea of who you are and where you're coming from to immunology and all the work you do now.
Yeah, no, I'd be happy to. I didn't have a classical route to immunology, as a lot of people do in the field. In college, I guess, I went to the town and the high school I went to didn't have a lot of science, and so I took the science courses that I could, and I liked them. And so when I went to college, I said, "Oh, I'm going to be an engineer," because that was like the only scientific career I even kind of knew about. And I like chemistry, so I was like, "Oh, I'll be a chemical engineer." And then I had to take, so I was taking all the kind of science courses that you would take for engineering, and I decided to take biology as an elective. And that was when I was first introduced into genetics and cellular biology, and I was just kind of blown away. And so I switched my degree at that time from engineering to cellular and molecular biology, and was working in a lab at the time this was at the University of Washington. I was working in a fly lab, but the fly lab actually studied human disease, and it studied a mental retardation syndrome called Fragile X. And so I was doing human genetics as my undergrad research, and that was really exciting. And so when I decided to go to grad school, I was going to go into genetics. I was going to get my PhD in genetics. And I started to look at some of them, rotate in some of the labs. I went to Stanford University for for graduate school. And then I started to learn much more about model organisms, because even though I was in a fly lab, in undergrad, we didn't really work on flies. We worked on human disease. And so I became very attracted to what I was learning about from my friends and colleagues that were getting their PhD in developmental biology. And so then I said, that sounds really cool, because they were doing all these really amazing things with worms and flies and zebrafish. And I said, this is cool. I'm gonna do developmental biology. So I started my PhD then in a developmental biology lab, and was working on, but I knew along the whole time from starting my PhD, the one thing I did not want to ever do is clone a gene. At that point, the genomes weren't sequenced. We were banging our heads against the walls. I saw so many PhD thesis projects get just like go nowhere for many years, because they were just trying to clone a gene. And so I decided I'm going to work on genes already cloned. And so I did, for my PhD work, I worked on basically more biochemistry and working on protein-protein interactions and trying to understand signal transduction downstream of the EGF receptor in C. elegans. So that was really a lot of fun. Learned a lot about developmental pathways. And as I started to think then about my postdoc, I knew that if you're going to pivot and do something different, your postdoc was a good time to do it. And of course, there was a lot of fascinating questions. The genomes were now getting sequenced in developmental biology. But I started to learn about what other people were doing and some of my other classmates were doing in the area of immunology. And there was an older student in the lab actually who was thinking about his postdoc, and he said, "I'm thinking about host defense." And I said, that's kind of interesting. So I learned a little bit more from him about what host defense was and started thinking about this. And then I started to go to the graduate student lectures that were being taught to the immunology students at Stanford, and sat down in some of these lectures, just listening to the ideas and the biology and trying to understand if this was something I would be interested in. And every lecture, I was just like fascinated. But I remember the one that Jerry Crabtree was teaching, and he talked about immunological memory. And that was the first time I had ever thought about immunity. Of course, I knew about like general aspects of immunity, but I didn't know that there were specific cells in our bodies that were formed. And so this whole process of becoming immune and this phenomena, I was just blown away. And from that point on, I said, okay, this is something that I would really want to work on. And so I was looking at postdocs and Rafi, and I also, because I had a PhD in model organisms and development, I knew I wanted to work on a model system where the pathogen was very well understood. The mouse host had the genetics that you could start to combine. So I found, I came upon Rafi and his work, and it was like, that was perfect. It was right. He was studying memory. He was studying model organisms and viral pathogens that had very well, great, a better understanding of, and now we're getting actually the epitopes mapped to study even the viral specific T cells. So it couldn't have been a more perfect timing for me. And so when I came to Rafi's lab, though, I didn't, I have to be quite honest, I barely knew what a T cell was. I barely knew that CD8s spoke to MHC class 1 and CD4s spoke to MHC class 2. That was like the minimum. That was like what my knowledge was. And then I came into the lab very green, but was fascinated by the complete black box of what we could start to ask as to how these memory T cells develop. So really, I brought a framework with me from my PhD to think about this problem of memory. And I think you can see some of those concepts and things that have decorated the way that we've thought about the problem over the years. So that's a longer answer probably than what you wanted to.
So go to the other end. So that was your origin story. I think given your body work and just how science goes in general, there's probably one or two moments you've had where you got the data and you're like, wow, okay, well, that was not what I expected, but also super awesome. So, and kind of one of those, phase shift moments that we all live for in science. So for immune memory, what are the top one or two of those for you over what you've found today that just like made you pause and like, the gears going, the light bulb shining, and then that exciting, like, oh, this is going to be big.
Yeah, I'd probably say one of the first questions that we really started to synthesize, I think, a lot of work in the field, but a lot of people were putting different models for how memory T cells develop at the time. We had the models where you come out of the thymus predetermined, you're going to go and become a memory T cell or not. We had models where there was a strength of signal model. We had models where there was a duration, a history of interactions with antigen, and then we had the asymmetric cell division model. And so a lot of these models were coming out, and there were models also that were starting to suggest that central memory cells, for instance, this is work from Sallusto and Lanzavecchia, who first helped us understand and really appreciate the heterogeneity of the T cell response and of the memory T cell populations, with the first real kind of identification of subsets of memory T cells. But within doing that and making those discoveries, they started to think about, well, how they form, and they were suggesting models where like you went from a naive cell to a central memory cell to an effector cell at the end, and that was really confusing to me. I think it was confusing to a lot of people in the field, because temporally, as we look over the course of an immune response from its start to its finish, we know that you are a naive T cell, you get activated, you differentiate into effector cells that then at the resolution of infection start to differentiate into memory cells. And so I think the temporal kinetics, all of that, but the naming of the cells was starting to jumble up create confusion. So I started to think about a lot of different aspects of what was coming out in the field, and then also thinking about some work where people were showing that if you stimulated T cells for a very long period of time, they could not become memory cells. But what I started to think about was really that T cells are differentiating on a spectrum, and that the spectrum is perhaps linear, maybe it's not exactly linear, but that these cells can take on multiple differentiation states. We weren't talking about states like we do today with single cell technologies. This was kind of a new idea to think about a state. We still were thinking about T cell lineages back then, but maybe as T cells differentiate, they can differentiate and acquire state that some will eventually become a much more stable, terminally differentiated state. But there's probably we knew already there's a whole degree of plasticity in T cells. And so maybe there's a lot of T cell states where T cells are kind of in limbo, and they can kind of move up and down. Some can certainly get pushed and driven to be more differentiated into a terminally differentiated state. But then there's cells that kind of sit in this limbo, and maybe even some can slide back and de-differentiate as well. And I think we started to think about it like that. And then we started to think about, well, what are some of the factors that may determine how cells fall on the spectrum, and thinking about inflammation and cytokines. And this really then was helped me think about some of the work that had suggested that a lot of inflammation or a lot of exposure to like IL-12 would drive T cells, they basically couldn't become memory. And I was thinking, well, maybe they're becoming these terminally differentiated effector cells, and that terminally differentiated state is what prevents them from becoming memory, but they are very productive, highly specialized cytotoxic T cells during the course of the infection, but then they lose their fitness, they lose their memory potential. And that was actually a concept that Rafi had first developed, that there's memory potential that cells have, and that can be lost. And so we started to kind of put this all together. And I think that then layering on top of that, that there's transcription factors that can guide this process, and perhaps even the amount of the transcription factor that's being expressed in the cell, that so the kind of the concentrations of transcription factors or their activities could make a determination of how far cells differentiated along these spectrums. And so I think that was kind of, to me, I think an important turning point, at least for me in the field, thinking about how you can basically provide an explanation for why there's so many different types of T cells, why some cells seem to have more terminally differentiated states and some cells have more plasticity, and then how you also layer in memory cell potential and how cells then may even lose memory cell potential. But of course, a lot of that was also helped with the leading from the discoveries of Lanzavecchi and Sallusto to now see there is more subsets within this population of CD8 T cells within this effector pool that could take on these range of states and fates. So I think those were probably some of the first biggest connections that we started to put together.
I guess this would be you're the right person to ask. So it is really difficult, and to also because much of this work has been done on the response to, for example, acute viral infections. And then you have all of these other situations, chronic infections, non-viral infection, cancer, and people are just trying to kind of apply this categories that were identified initially in these models, and then see, well, you have these effector memories in tumors. So let's just take a step back and think. Currently, what is our best understanding in the case of a quote unquote straightforward acute viral response? What is our understanding of the priming, the effector populations, and the origin of the memory, the long lived memory cells that we have now after all of these doubts, all of this research? Where are the memory cells coming from, and how do they replenish the second line of defense?
Yeah. So I think we know part of the answer to your question to a reasonable degree when we look at systemic infections, right? So I think that's a lot of what the basis of the work has been on as well. So these acute systemic infections, like we have with LCMV or Listeria infection. But then, of course, what we have learned over the last 10 to 15 years is how much of the memory is in our tissues, right? And these long-lived tissue resident memory T cells. And we're learning also from Dave's lab that they're not, some cells do kind of come back out of the tissue and join circulation or hang out in the draining lymph node. But I think if we just kind of keep it simple, and that we've got long-lived tissue resident cells, and then we've got circulating memory cells. So I think we really started to dissect and figure out kind of the precursors that we can identify within a systemic in the circulating pool. So we were able to, as we kind of looked at the spectrum, we were able to find cells that didn't fully terminally differentiate into highly polarized, specialized cytotoxic T cells, but they kind of maintained this plasticity. They maintained multipotency to give rise to memory cells. And epigenetically, this was really fun. We were able to show that these cells, these that we call memory precursor cells, despite they would maintain their epigenome, they would maintain the gene expression the loci that would be expressed in terminally differentiated cells and memory cells, they kept those genes open. They just weren't transcribing the cells, the genes that were involved in driving cells towards terminal differentiation. So you could see how they were mechanistically, you could see how they were maintaining this multipotency and this plasticity. They kept the genes open that they could then transcribe at a later state. Whereas these terminally differentiated effector cells, they started to shut down and silence and repress the loci that were found expressed in memory cells. So we saw that these cells were losing their memory cell potential by essentially shutting down memory genes that were expressed in memory cells. So I thought that was a really interesting understanding now of how memory cell potential is being regulated within these cells. So then these memory precursor cells, they certainly have the capacity to, some will die over the course of the resolution and the contraction phase, but they do have the capacity to seed a lot of the circulating memory T cells, both central and effector memory. So, but the tissue resident memory cells, I think we're still not entirely clear on where the progenitor cells for those cells emerge. We know that cells are entering the tissues within the first few days after an infection. We know many of those cells are going to stay there after the infection, but we don't know, I think, well enough yet where those precursors for entering the tissue and becoming a tissue resident memory cell, where they're emerging. Are they emerging from a broader pool of precursor cells that give rise to circulating memory and to tissue resident memory cells, or are they cells that migrate into the tissue, inhabit it, and then receive cues to tell them to stay preserved as long-lived memory T cells in those tissues? So I think that's still a big question that we don't fully understand yet.
So to segue into some more recent work, you have a really cool paper out pretty recently on some, I'll call it neuroimmunology, right? Linking synthetic nerve drive to T cell exhaustion, which makes a lot of sense for anyone who doesn't even know biology, right? Like a lot of adrenaline, you eventually get tired, but it's obviously possible. So if you could talk a little bit about that, and maybe even if you can, what some of your follow-up work is on that angle.
Yeah, so this was a really new venture, an area for our lab to move into. The postdoc who has done the work is Anna Maria Globig. She'll be starting her own lab at the Salk Institute very shortly, and she was the first to really keen in on by looking at the genes that are expressed by exhausted T cells, and notice that these adrenergic receptors that were being expressed on exhausted T cells, but also on kind of more some of the effector-like T cells that we also see in chronic viral infection, and really started to think about why would a T cell be talking to a nerve? And this was during COVID. She had just started in the lab actually, and then the lab shut down. So she was like, "Should I pursue this?" And I was like, "Yeah." I go, "This sounds interesting. Let's check it out and see what we find." We weren't really thinking about at the time about. I guess we were thinking more about adrenaline. That was kind of more what I was aware of. Like there's adrenaline. You get stressed out. You have a shot of adrenaline go through your body. And so I was thinking more probably that would be. We weren't really thinking that when we started this project about how the tissues are innervated by our peripheral nervous system, how then when we think about where T cells are within the tissues, how are they then possibly communicating with peripheral nerves? And so that really, I think, to me, opened up a whole new way of thinking about this of tissue biology. That, of course, we think about all the immune cells that infiltrate tissues. We think about the endothelium. We think about fibroblasts to a certain degree. We think about the parenchymal cells in different tissues, but I hadn't really appreciated and thought about nerves. And so that became a new way for us to think about it. And I think probably one of the most interesting parts of that story was learning that T cells could talk to nerves through adrenergic signaling is by far one of the most important discoveries of that work, but also just that they do physically interact and locate. So the T cells are actually migrating near the nerves, in at least in these tissues that are chronically infected, and we can see in some of the tumor models we've looked at. So there's actually a chemotaxis. Well, we're assuming there's a chemotaxis of the T cells to the nerves, and there they receive signals that then shut down their responses. Now, you could think that this makes perfect sense because our nerves can't get attacked. You don't want our nerves to get attacked by our T cells, since they do have difficulty regenerating themselves. That would make a lot of sense. So that's also opened up a whole nother idea. It's like, well, how then is the environment around nerves and tissues and different types of nerves could be modulating the immune response locally, and how far out does that spread from the nerve? How far out does the noradrenaline get secreted? I think there's a lot of really interesting questions as it relates to how the nerve composition and density and types of nerves and their activity then regulating the local immune cells around them.
I guess I think there's been lately also when we go to conferences and the latest papers, this has been a very much growing area, the
Mhm.
interaction between T cells and immune cells, but I think particularly T cells and nerves. I think it's absolutely fascinating. One of the things I keep thinking about, especially when it's about this kind of stress associated, is this are these mechanisms by which being stressed is making you sick? Is this part of
Yeah,
that?
that's what we don't know for a fact, but chronic stress is definitely associated with a lot of comorbidities, and immunosuppression is one of those. And so it's possible. I think we need to expand our range beyond exhausted T cells and think about how neurotransmitters are being affecting different types of immune cells and also different types of T cells, but over the courses of different types of immune responses. So we don't really know. And we also don't know. Think about these nerves are sitting inside these tissues. Now there's an infection going on. We don't know how the nerves are responding to the inflammatory mediators that are being produced locally. So, and then how is that going to be sent back up to the brain? And how is the brain going to come back and tell the nerves how to react in that tissue? So it's opening up a whole ability to think about the body to brain axis and the circuits that truly are being activated by an immune response occurring in a peripheral tissue.
We know that some endogenous opioids and exogenous are immunosuppressive in long, in multi-dose scenarios. So that would imply like chronic pain also could be immunosuppressive, which goes along with stress and everything else. So yeah, it's super interesting. I wonder, do you think we have the model systems today in a way we didn't to be able to answer some of this? Do you think that's one of the big changes? We now have the tools.
Yeah, yeah, and also getting neuroscientists and immunologists to talk to one another. That was very clear at the I was just at the Neuroimmune Keystone meeting last week, and it was very fun because you're going to it reminds me back when I started as a postdoc in Rocky's lab. You got someone who's never even thought about these areas of a field talking to each other and really and teaching each other basic concepts, right? That they can understand how the immune system's functioning, or we can understand how the nervous system's functioning. So yeah, I think it's a lot of fun to get two people into a room that have never really thought about each other's systems and how they could be really interacting. So
Yeah. I mean, I think probably neurobiology or like neurosciences is the one other discipline that is just as complex and just what is the word? Just like brain rackingly difficult,
no
like
pun intended.
immunology. Yeah, exactly. No pun intended. But I think that's kind of like this is two disciplines that are already huge on themselves, and but there's so much nuance when you start finding these connections. I think the first story that I heard, I think it was some time ago, about inflammation in the brain causing psychosis or like psychological symptoms. And I think then we have never stopped realizing that, of course, they are interacting with each other, and not surprisingly, right?
Yeah.
Um,
Yeah, I think a lot of mental health in diseases, schizophrenia, depression, when there's already clear correlations of changes in immune cells and inflammatory cytokines, but we don't know enough yet of the mechanism to how they're causal. But I think that's where I think we'll start to understand a lot more of how our immune system is modulating behavior and mental states.
Yep.
I'm very excited for that area. And I think also, Jason, you'd asked like kind of where have we gone from some of this work, and it's really started to make us just think a lot more also about moving away from the peripheral nervous system but to the central nervous system, and what happens after an infection to immune cells in the brain. Where are they going? Where do memory T cells in the brain live and reside? We know we have long-term resident memory T cells in the brain. Where are they? What do they do? How do they contribute to age-related inflammation that's associated with a lot of these neurodegenerative diseases? And it's been fascinating because the field of Alzheimer's disease and also Parkinson's disease has been finding, contributions of T cells in these diseases. And so we're also very excited about trying to tease apart, well, which types of T cells are involved in these types of pathologies, and how much of some of these neurodegenerative diseases. Parkinson's clearly has an autoimmune component, at least in some individuals. How many other neurodegenerative diseases will be due to an autoimmune-like component? So, yeah, I think there's going to be a ton of exciting things coming up.
I think is very, very interesting. I just wanted to quickly touch upon, I thought it was also very interesting, the talk that you gave at the AI this year. You were talking about, so some of your recent work has looking also into metabolism of T cells and particularly some lipids. And you had a really cool story about bile acids and how, if I understood correctly, I'm remembering now, they interact with the microbiome that is, metabolizing these bile acids, and that has a surprising effect on T cell immunity. So I was wondering if you can just give us a quick overview about that story, because I just want the listeners to hear it, from you, uh, because I think again another level of complexity that it's just shows how much everything is connected, and I really like that story too.
Yeah, thank you. This is work from Karthik Varanasi. He's a postdoc in the lab. He joined the lab because we've been interested in extending from this idea that we know immune cells are regulated metabolically and that their function is going to be regulated by certain metabolic pathways that they're using and which nutrients they're using. And so we started to think about, well, when they enter, especially these tissue resident memory cells, or when T cells infiltrate tissues, they're now engaging in a different metabolic landscape that they had before. If you think about it, it's really fascinating, right? These naive T cells that are circulating through secondary lymphoid organs in the blood, they then get activated and they go into a completely different environment. And they had never been educated on how to live and adapt in that environment. And so that's something we were thinking about. It's like, well, how do T cells adapt to the tissues? And are there going to be metabolic cues and aspects of different organs that one might need to be thinking about that the local T cells, the resident T cells, are starting to adapt to? And we've been focusing on this question mostly in cancer because cancer is usually, it's a metabolically dysregulated cell state, as the tumor cells need to become very anabolic to grow and to become evasive and to metastasize. And so how does that metabolic perturbation occurring in the tissue then change the way in which the immune cells are locally affected by what metabolites and nutrients are available? And so we started to say, well, let's kind of put these two ideas together and look in the liver, since the liver is a very metabolically active organ, but also has performed some very important functions that are rather unique to the liver. And so bile acids was one of the lipids that we started to think about. How could that be modulating local immune cell and T cell function? So my postdoc started to look at in liver cancer models at the production of bile acids. We found that they were elevated in these livers that had cancer, tumors growing in them. And then we found that the T cells actually that were recognizing the tumor were able to import more bile acids. We could find more intracellular bile acids in those T cells that were recognizing the tumor, but we were able to compare that to T cells that were just sitting in the liver because they were resting memory cells from, say, a prior infection. So using this model that Andrea Scheitinger had developed, where you could look at memory T cells to one pathogen versus T cells to another antigen, in this case, a tumor, you could compare them in the same liver. And it was really interesting because the tumor reactive T cells were importing bile acids more so than the resting memory. That made me make sense because you got antigens that they're engaging, they're definitely going to be in different activation states. And so, but then it would be made us start to think about as well, how do the T cells detoxify themselves from these bile acids, since when you accumulate bile acids, it can be very toxic. They're basically detergents. And so how does that then change the way the T cells thrive in this environment? And so Karthik found that the T cells definitely are less, the T cells that are reacting to the tumors are less able to efflux and get rid of these bile acids, and that's why they're accumulating. And so if you're able to manipulate their ability to efflux the bile acids and enhance their ability to get rid of these bile acids, you can enhance their survival in the tumor and enhance their function. So that was kind of one aspect. But then kind of what you were asking about as well, the microbiome will then metabolize these primary bile acids and turn them into secondary bile acids, of which there's hundreds, thousands of secondary bile acids are being produced by our microbiome. And we found that some of these secondary bile acids, we've only focused on a couple, are able to also have effects on the T cells in the liver that can either enhance their activity or suppress their activity. So there's going to be probably quite different depending on the mixture of secondary bile acids that the cells are being exposed to. But one thing that you can do is therapeutically, you can give secondary bile acids in the diet. And so if we can learn more about what these secondary bile acids are doing, we're able to find that some are going to help enhance immunity and some are going to actually suppress T cell function. So we're very excited about kind of playing around and dialing up and down immune responses with these secondary bile acids. So
I would go very deep into the microbiome bile acids field, but we will run out of time here.
And good, because I don't know. That's all I know.
I work at a microbiome therapeutics company, so
That's all he talks about all the time.
all the jokes, all the time.
Yeah.
That being said, we always like to end up with a science adjacent question, if you will. So if you were not a scientist and you had gone down a different route or chemical engineering, what would you be?
I have two answers for that. I love plants and I love flowers. And so one job would be to be a florist and just make beautiful bouquets all the time. Right now the jasmine and gardenias are blooming, and it smells amazing around here right now. But probably that wouldn't satisfy me on a everyday work schedule. I actually think, and maybe it's just because I watch too much binge watch too much TV. I actually think I'd probably be a detective, and so I think I would like CSI. I think I could be like one of those people. But I hate guns, and I would never want to hold a gun. I'd never want to shoot a gun. So I don't know how good of a detective I would be.
Have to go to the UK or something.
Yeah.
Right. Yeah, they just have batons and knives.
Yard.
Yeah. I was scared for a second and said, I'm watching too much TV. I hope you weren't going to say, I wish I were a desperate housewife, you know?
More Hieronymus Bosch or something.
Yeah.
All right. Well, thank you for coming on. This has been an absolute pleasure.
Thank you. Thanks
Indeed.
for having me. Yeah.
So thank you so much. We had Dr. Susan Kaech from Salk Institute. It was awesome having you around. Thank you so much for joining the podcast today.
Okay. All right. Bye-bye.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.